So the second part of this lecture is actually showing you the results of the Gloria trial, which I think is the solution to the problems of confounding by indication. And before I start, just to show you what a project like this means in time, I think we took about 10 years to get this study done. So it's doing a trial is not a decision to take lightly. So I have to thank all these people, of course, the patients, the people in the core team, the partners of the project, the clinical centers, and I think the advisory committee is many of those. For me, at least, and this has been my part of my life work. People often say, "Why, these trials they only give limited information because they're and the perception of selected populations that are much more healthy. This is a normal drug. So results of the trials not relevant to me. I need real world data. If you give it in high doses, it is very, very doing a randomized trial. That is a problem of the design of the trial, where the designers and select a very specific this is like any other drug. And given the fact we should start looking at it as a normal drug, and in RCTs, randomized controlled trials on drugs are being performed by the pharmaceutical industry, preventing harm from the drug have a very specific intent, which is there's no time to go into that, but document data which allow the industry to get the tool registered, used. And it should be used wisely. And if we in humans, and that is a very specific placing the glucocorticoids in a separate objective of those trials, which means tell each other healthy populations they don't want those possible pure the lowest drug result because to be spoiled by all the other things that go on. This is not how we work with drugs, because in effect, patients, in most practices, and probably you should not, if you were to blame that on you are giving it for longer periods, randomized design, then you should blame it on the selection in more people than you think. Now the Gloria trial was a pragmatic trial. Now this trial shows, so it was a real trial, placebo-controlled, but it was pragmatic, which meant that we maltreating specifically looked and you are not causing that undue ones you see in your regular compared to the other treatments. And that is the introduction. So that's why the data of this trial is so important. No drug is 100% think about the benefit and harm of glucocorticoids. It's really cool to see that even with this low dose, you can help patients. This is the evidence that's already out there. If you look at some carefully these are the disclosures, like the and also trial, just to remind you that I wrote this book that you should buy minimal disease activity on tocilizumab. It's and 5 milligrams. This link alone. If you take the prednisolone away, and you should be aware that this full study report of the Gloria trial is already published you that in ARD, and it's open access, which we know is a powerful drug. And there is something to be added by giving prednisolone. It was funded by the and if you give it in patients who need it, Horizon 2020, outside of a program, you're giving it to high-risk patients. So you should expect more harm. It was not funded by any because they're high-risk patients. Funded, so you should address the harm by looking carefully. The background is that many RA patients are on chronic prednisolone at their blood pressure, advising against all the other stuff. The objective of the study was to compare 2 years of prednisolone, that's 5 milligrams per day, or placebo, added to optimum standard of care in RA patients aged 65 or over. Now, the requirement for 65 or over was from the EU because their funding program wanted trials in the elderly because the elderly are underserved in terms of data generation. And for us, of course, it was a way to do our trial, even though our real study question was not limited to elderly patients. On the other hand, doing it in this population increased the a priori chances of harm, because obviously there's much more comorbidity in that population. And also, low-dose prednisolone is quite often given to elderly patients because of the perception, right or wrong, of physicians that the low-dose prednisolone is less harmful in this group of patients than the biologicals and other treatments that we use for RA. Anyway, that's the way it happened. So it was a pragmatic double-blind trial, as I said, which meant otherwise, from the age criterion, there were minimal eligibility criterion. And also, very importantly, all co-treatments were allowed. Now, in registration trials from industry, co-treatment is fixed at baseline, and you cannot change anything during the trial, because if you do, you will spoil your treatment contrast. And we allowed all treatments because we felt, if in real life, patients get all kinds of things if your initial treatment doesn't work. And we did not want to limit the physicians to treat their patients as they thought best. Of course, with the exception of chronic low-dose prednisolone, which was the study intervention. So physicians were not allowed to do that. And if they did, the patient was taken out of the trial.
I have just one
yes?
commentary about the pragmatic trial. Usually when we were students and we heard about pragmatic, it sounds like it's more robust than a real RCT. But it's a real RCT also, even if
yes.
it's a pragmatic.
Yes.
Could you just please explain this this notion?
So in the terminology, pragmatic means, as I explained, close to the regular, as close as possible as you can get to the regular situation in terms of population and treatment. It does not mean that the trial methodology itself is changed. So it's real randomization. You have to have a real control group. In our case, it was placebo, but you can also do a randomized control trial with 2 active treatments being compared. In our case, we added our study treatment to whatever the doctors were giving for the rheumatoid arthritis anyway. So even though it was a placebo control, the placebo group got the best treatment the physicians could give, except for study treatment. So pragmatic is really about the applicability of the study results in terms of population and other treatments.
Okay, thank you.
Yeah?
Yes.
So then as benefit outcomes, we use the DAS28 and the Sharp-Van der Heijden joint damage score. And as harm outcome, the primary outcome as defined was the proportion of patients with at least one adverse event of special interest. And the adverse event of special interest was defined as either a serious adverse event, which has a very specific definition in good clinical practice guidelines, and also a group of glucocorticoid specific events that we defined before the trial. And finally, all events causing study discontinuation. On the next slide, I will show you the glucocorticoid specific events. For analysis, we used longitudinal models, and also important to say, we used one-sided testing and 95% confidence limiters. And why did we do that? Because we had very strong opinions, expectations on that we would see more benefit, but also more harm. So two-sided testing was not appropriate. And because we did one-sided testing, we had more power in our trial to detect both benefit and harm. And then because we had 2 primary outcome groups, so we had benefit and harm, which is also different from most trials, where the trials have the primary outcome on benefits and then they report the harms. We said, no, we want our trial to look as outcome at both sides of the coin. So we had predefined decision rules to decide whether our treatment was a success, whether it was a failure, or whether there was trade-off. Now, what are those glucocorticoid specific adverse events of special interest? We define that as myocardial infarction, cerebrovascular or peripheral arterial vascular event. but also newly occurring hypertension, diabetes, infection, cataract, or glaucoma requiring treatment, symptomatic bone fracture, and all these events were adjudicated on the blinded data. So before we removed the blind, we decided whether an adverse event would be coded as adverse event of special interest or not, based on physician report and extra information that we gathered on queries. So the results. We included the patients in 2.5 years, a total of 451 patients from 28 hospitals in 7 EU countries. And we were able to include almost all of them in our safety and efficacy analyses. Patients had an age of mean 72 years, had established impactful RA, as shown from the mean DAS, the HAC, the percentage of patients with joint damage, and most of them were on disease-modifying treatment, and 14% were on biologics. And also they had a median of 3.5 active comorbidities. And I have additional information in the backup slides if required. 62% of patients completed the whole 2-year study period. And the reason for discontinuation was in majority other reasons, including COVID-related reasons, especially in the second part of the trial. We had a lot of problem getting patients to come to the clinic for their especially final visits. So we had a lot of dropouts because we could not collect the information because patients were not able to come to the clinic. And of course, we had patients stopping for adverse event and lack of efficacy. And the proportions were equal in both treatment groups. Mean study time in study was 19 months. On the benefit side, the mean effect of prednisolone over 2 years was a DAS28 of 0.37 points lower, and for joint damage progression, 1.7 point lower, and both effects were highly significant. And pre-specified secondary analysis suggests that there was more benefit for prednisolone in first year and less in the second year, and that there was confounding due to co-intervention. Remember, we allowed all co-interventions, and those confounding, the co-interventions were benefiting placebo more than prednisolone, so they had the effect of reducing contrast between the treatment groups. Especially, there were significantly more placebo patients with a treatment switch, and subsequently disease activity likely decreased. And there was a trend towards more prednisolone patients tapering treatment, which subsequent expectation that the disease activity would then after that tapering increase again because of a flare. And these analyses, these confounding analyses were also predefined in the protocol. They were not post hoc. So this is the graph of the main effect in the primary model, where we modeled the mean effect of treatment over the 2 years. And you can see both groups have decreased disease activity in the first 3 months. And then it sort of stabilizes after 1 year. And the gray area is a graphical trick that allows you to see where the two treatment groups would be if there was no significant difference.
Okay.
Now, we did, in secondary analysis, we did also a model with time treatment interaction, as I already mentioned, and that's the second panel. So the first panel is the one I showed in the previous graph, and the second panel below it is the model with time treatment interaction. And there you can see that the contrast between the groups is larger in the first year and then smaller in the second year. And then finally, the lowest panel is the data as observed, which is obviously disturbed by the fact that people dropped off. So they were not in the group anymore. So the means that you see on the graph are not from the same group of people. We also did, as a sub-analysis pre-specified, something we called the pure treatment comparison, to sort of emulate a short-term placebo-controlled trials. So we looked at the data of the first 3 months in a subgroup of patients we called the per protocol patients, and those patients were patients who did not treat switch their treatment at baseline, because we allowed that, so patients could go in the trial and be started on a biologic, for example. And obviously, starting on a biologic will have huge effects on the initial decline in disease activity. So if we take those patients out, we take the patients out that had glucocorticoid injections in the first 3 months, and we included only patients who had at least 80% adherence on study treatment, full assessments, and no other protocol violations. Then we are left with about 300 patients equally distributed over the 2 treatment groups. And then the DAS28 is 0.62 points lower on prednisolone, obviously highly significant. And if we look at this, the response indices like the EULAR and ACR response, you can see that the blue groups, light blue groups, which is prednisolone, have a lot more good responders. And in the ACR 20, 50, 70, you can see that the blue columns are higher than the dark blue columns. And finally, if you look at disease activity state, minimal disease activity, a lot more patients in the prednisolone than in the placebo group. And even there is a difference in patients reaching Boolean remission after 3 months, although the proportions, of course, are very small. We're talking about 5 milligrams of prednisolone, remember?
Mm.
So that's a low dose. Now, let's turn to harm. There was more harm in the prednisolone group. 60 versus 49% of patients had at least one adverse event of special interest, which boils down to an adjusted relative risk of 1.24, with the 95% confidence limit at 1.04, which is significant at a one-sided p of 0.02. The largest contrast in events was in mostly non-severe infections, and the adverse events clustered within patients. Other glucocorticoid-specific events were rare. There was no clear signal in cardiovascular disease, diabetes, or bone. And the table shows you the primary outcome and the distribution of that over patients with only severe adverse events, only other events, and a combination of the two. If we look at the harm by organ class, and now the numbers are events per 100 patient years, you can see that there is a slight increase in many of the organ class groups for prednisolone, but the only real increase you can see in the infection row, where the numbers are a lot higher in the prednisolone group than in the placebo group. There were a total of 5 deaths, 2 in the placebo group on treatment, and they were both cardiovascular, and 3 in the prednisolone group, 1 on treatment, and 2 just after treatment within months after premature discontinuation. So to conclude, add-on low-dose prednisolone in established RA has powerful long-term effects, with a trade-off of 24% increase in patients with mostly non-severe adverse events. These results are immediately applicable in clinical practice and suggest a favorable balance of benefit and harm. So, and I think the key is, for me at least, and this has been part of my life work, to try and turn glucocorticoid and the perception of glucocorticoid back to this is a normal drug. So it is not a miracle drug, although if you give it in high doses, it has very, very powerful effects. But it's a normal drug with advantages and disadvantages. But this is like any other drug. And so we should start looking at it as a normal drug. And in the right hands, and I think the rheumatologist is the right hands, because we perform a lot better in terms of preventing harm from the drug than other specialities. There's no time to go into that, but in the right hands, this is a tool that should be used, and it should be used wisely. And if we keep on placing the glucocorticoids in a separate category, which means we tell each other we use it in the lowest dose possible for the lowest period possible because it's so dangerous. But in reality, this is not how we work with the drug, because in effect, in most practices, and probably even in yours, if you were to look, you are giving it for longer periods than you think you are, in more people than you think you are, because that's the reality. And now this trial shows that if you work according to that principle, you are not maltreating your patients, and you are not causing undue excessive harm compared to the other treatments you give. But it's not 100% safe. No drug is 100% safe. But it's really cool to see that even with this low dose, you can help patients, and it adds to the evidence that's already out there. If you look at some carefully performed trials, like the Samira trial, where patients who were in minimal disease activity on tocilizumab and 5 milligrams of prednisolone, if you take the prednisolone away, a substantial proportion of people will flare. Which is telling you that even if you give tocilizumab, which we know is a powerful drug, there is something to be added by giving prednisolone in patients who need it. And if you give it in patients who need it, and outside of a trial, you're giving it to high-risk patients, so you should expect more harm because they're high-risk patients. And so you should address the harm by looking carefully at their bone, at their diabetes, at their blood pressure, and all the other stuff, the infection propensity. Yes, that's what you should be doing.
Yeah. Thank you. Thank you, Prof. Boers, for this amazing talk.